So these are all sea urchin embryos. So, I mean, sea urchin eggs, sea urchin embryos. And the only reason why I'm using these is to show you that it doesn't matter what species you're in. The names are the same. So when you have, here's an 8-cell embryo, and each one of these cells is a blastomere. And these blastomeres, these are the totipotent stem cells. Okay? So if I took each one of these blastomeres and put them in a centrosoma, I could create a complete, intact, normal individual. We can do this with humans too. We can clone everybody during this stage of embryo development. Okay? So these are blastomeres too. I think this is a 64 cell. And eventually it metamorphosizes into a gastrula. This is the completion of that stage. And I'm not going to ask you anything, Harry, other than know the difference between a blastomere and a blastocyst. And we'll come into that in a moment. So this is a germinal vesicle embryo. This is what you ovulate. There's the nucleus this is what you ovulate. This is the egg you ovulate, not the embryo. This is going to be an embryo, provided fertilization is successful. So the bosona pellucida is right here, slightly out of focus, but you have all these granulosa cells out here. Okay? And they represent these specialized granulosa cells. The ones that are stuck to the zona pellucida right here are the corona radiata, named after the corona of the sun during a full eclipse. And then the rest are the cumulus oophorus. So those are specialized granulosa cells. They're shown here too. But the nucleus is out of the plane of focus. It's right about here. And the zona is there as well, slightly out of focus. But all those granulosa cells accompanying the egg is what you ovulate. So we've seen this image already, okay? Pronucleate, embryo, viable. The other one is not. And here's how we manipulate them. So you can actually remove a pronucleate or put something else in there. I don't know what they were doing in this particular experiment, but this is how you do it in the lab. You actually make these microneedles and this micropipette that creates negative pressure that holds onto the egg so you can manipulate it. So here is a egg that's been in culture way too long, and it literally looks like a fried egg. And so this is dead. Eggs and embryos don't like to be in culture. Here is approximately an 8-cell embryo, and each one of those cells is a blastomere, as opposed to a blastocyst. This whole thing is a blastocyst. Okay? So this becomes a 16 to 32 cell embryo, and when it's doing that, it's expressing embryonic cell adhesion molecules. And what they do is they bind to each other's cytoplasm, and you lose the delineation from cell to cell. So even though this is more cells, you can't see very many cells because of these cell adhesion molecules compared to this. Because this is 8 cell, this is 16 to 32. And when it's from 16 to 32 cells, that's when the first differentiation occurs. The first differentiation into what you call a morula to a blastocyst. Okay? So here's my blastocyst. And the blastocyst basically consists of the inner cell mass and the surrounding trophectoderm. So during the morula stage, the inner cells of the morula become the inner cell mass, which is destined to be the embryo. The outer cells of the morula become trophectoderm, which eventually becomes the placenta. And it's this trophectoderm that we'll be talking about later. They're responsible for the second recognition of pregnancy. We'll deal with that later. So just remember, trophectoderm, second recognition of pregnancy, human chorionic anatomy. We'll deal with that later. Now, there was an experiment done where They took the cells on the inside and put them on the outside, and the cells on the outside and put them on the inside. And guess what they got? A normal embryo. The cells that were placed on the inside, but originally from the outside, became the inner cell mass. The cells that were originally on the inside, placed on the outside, became placenta. So, what's happening here? These cells in the early embryo are position sensing. They sense which position they're in by touching each other with the cell adhesion molecules, and they differentiate along that cell line. If they're on the inside, they're going to become the embryo. If they're on the outside, they will become the trophectoderm, destined to be the placenta.
What happens if you do that after a certain point, though?
Well, you can only do it during the memorial stage and get it to do that. Okay? These cells right here, the inner cell mass, right here, these are still pretty much totipotent. In other words, any one of those cells can become a complete individual. Okay, so these, an 8-cell embryo, these are the embryonic stem cells that everybody talks about. Media. These are it. 8-cell embryo. Okay?
What does the blastocoel cavity?
The blastocoel cavity,
What does that become?
it's just a fluid environment, and nobody knows why it's produced or basically why the embryo does this. Okay? So the blastocoel cavity, now imagine this blastocyst, and don't confuse blastomeres with blastocysts. Okay? Blastocyst is the entire embryo. And they make a blastocoel cavity, and we don't know why. It has to do with some sodium pump, sodium-potassium pump, probably. Pumping sodium out and water accumulates. Okay, that's what's happening, but we don't know why. But you got to imagine that in this light microscopy, you're taking light sections of the embryo. So the reality is this blastocyst is really a ball with a ball in it. The ball is the outside trophectoderm all the way around, and the inner ball is the inner cell mass, destined to be the embryo proper. And in that process, you're making fluid and creating this blastocoel cavity. So here's an embryo in different cultures. These are all embryos I put in culture, so these are images of my culture. So this is a 4 to 8, and then here is an early blastocyst. You can see the blastocoel cavity here. Here's a nice zona pellucida. And then this is the same as this, which is the same as this. Now you have in culture, the embryo expands, expanding this blastocoel cavity, putting pressure on the zona, and then eventually the embryo releases some enzymes that breaks down the zona pellucida and the embryo leaves. Okay? Now, this is not what happens during pregnancy in the individual. The embryos that get out of the zona pellucida look more like this one right here. Development is delayed in culture. So it takes longer for development to occur in culture than it does in the intact animal. Because we haven't figured out what are the exact media conditions that are necessary for normal embryo development. So this right here, this expanding and contracting like this did here, and then releasing of enzymes, are probably implantation enzymes, not zona escape enzymes. So it's different. So these are the same cultures, and you can see when, there's the dates when I made them, and times. So this embryo right here is the same as this one, which is the same as this one, which is the same as this one, and literally, when they're getting out of the zona pellucida in culture, they're literally walking out. You can see them with these micro appendages called trophectoderm projections, literally crawling out of the zona pellucida. So these embryos in early development are able to find where they want to go and using these projections to implant life, which is really, really weird. Okay? So I wish I had the movies. Maybe I'll try and get some again sometime. So here's an embryo that lost its zona. some with still zona intact, and that's it.